All right, 3 o'clock. Welcome everyone. Um, I'm going to read the Yolo County land acknowledgement and then we'll get started. We should take a moment to acknowledge the land on which we are gathered for this meeting. For thousands of years, the land has been the home of the Patwin people, including the Yocha Dehe Winta Nation today. The Patwin people have remained committed to the stewardship of this land over many centuries. It has been cherished and protected as elders have instructed the young through generations. We are honored and grateful to be gathered here today on their traditional lands. Please welcome Chris Golé, who is going to speak about mathematical patterns in plants. Chris is at Smith College on the East Coast right now, so this is super exciting. Um, and please put your questions into the chat and I'll read them out periodically. And lastly, I want to let everyone know that our next workshop is Thursday, February 12th at 3 o'clock, and the topic is spring into irrigation. Okay, Chris, it is all you.
Thank you, Joan, and thank you for the invitation. And it's fun to be talking to people who are interested in gardening. My wife just finished the Master Gardener training in um, in December, and in fact, she's going to her graduation tomorrow. She's baking brownies for that. So I know how, for those of you who have gone through the training, who are going through the training, how rigorous it is. But I'm hoping to make this lecture not too hard and not too technical. So, it's weird to think that plants may be um, related to math, but there is a lot of math, and it's been fun for me to discover that. So let me get on with it. So what you see here on my first slide is a cover of my book. Here it is. Same thing. No, it won't. It won't. Yes, kind of. Um, so it's a book that I wrote with three other people, and I will let you know who they are and how that it happened. But it's a book that tries to be historical, to be really for a general audience of non-mathematical minded people or even scientific. We hope to also appeal to gardeners and to the humanity behind the science. So one of the main themes that recurs in the book and in my research indeed is spirals in plants. And the cover, we could not resist the cover, but this is not the kind of spiral that we're looking at. This is a beautiful fiddlehead, but this is not the kind of spiral I will be talking about. So what is it that I'm going to be talking about then? So it's this kind of spirals. I'm curious, do you see my mouse when yeah, okay, cool. So here's one spiral on this pine cone, for instance, but you see there are several going in this direction, but they are also going in that direction. And you see them a little bit less maybe on this Aeonium, but it's still there. They're still spirals, and they're pretty clear on this Dahlia. So one thing's one thing that people started doing in the early 1800s when they really kind of made it conscious to their mind that, these are spirals, why don't we count them? So they started counting them. And in the examples that I'm showing you, the pine cone has eight going in that direction. We'll say that this is counterclockwise. And the dahlia has 21 and the aeonium has two. Okay, so big deal. So then you count the other, the spirals going in the opposite direction, this time clockwise, starting from the center. And this time we have 13 for both the dahlia and the pine cone, and 3 for the aeonium. So what's the big deal? The big deal is that all these numbers are part of the Fibonacci sequence, right? So you see that the 2 and 3 for the aeonium are there, the 8, 13 for the pine cone, and the 13, 21 for the dahlia are all part of this Fibonacci sequence, and I'm going to explain what it is in a minute. But note that also those two numbers for each plant are successive members of that Fibonacci sequence. For instance, for the dahlia, 1321 are consecutive, and for the pinecone 813 are consecutive, and again for the aeonium 2 and 3 likewise. So the Fibonacci sequence, what is it all about? So it came about this fellow Fibonacci, Fibo as in son of, not Phi as son of Bonacci, the name of the family. And he introduced the Arabic numerals or the Hindu Arabic numerals in Europe in the year 11 something, close to 1200. And and for practice, he gave exercises for people to practice with those numbers because they had been doing computations with Roman numerals, and that was not easy to multiply Roman numerals. So he was giving them fun problems, and one of the problems was a completely fake problem about rabbit reproduction, and the solution to this rabbit reproduction problem was, how many pairs of rabbits do you have each month? If every 2 months a pair of rabbit begets another pair of rabbit, and the solution he gives in very great detail is this sequence. And he had no idea about plant spirals, and that was not his idea to relate them. And it's only at the end of the 19th century that somebody said, "Oh, look at this! These plant sequences, they actually appeared in this old manuscript by Fibonacci." So there we go. That's how it got its name. But Fibonacci knew nothing about plant spirals. So the occurrence of these Fibonacci number is not it's not an accident, and it occurs so often that you cannot not know it, and you cannot you know uh, not think about it once you've been exposed to it. And so many people from many different fields, mathematician, physicists and biologists and computer scientists have been interested in this. And everyone, including myself, says, oh no, this is the reason why the Fibonacci numbers enter in plants. So I will give you a little taste of my version of that. Um, so this is the main story in the book, and there are other stories that I will touch on at the end of the talk. Um, in the book, we have those pictures in the middle of two chapters as in transitions, and just, you know, gorgeous pictures. And we found this fellow who is a photographer, plant photographer, mostly cacti in Texas. And we contacted him and said, we love your pictures. Would you be willing to do a few pictures for us? For our book. And this is one of the picture that he did, but he gave it to us too late to include in the book, but I love it. And this is, as you might know, a broccoli romanesco. And the beauty of it is twofold. Not only does it display, I believe, 13 and 8 spirals, like the pine cone earlier, but it has spirals within spirals within spirals within spirals 7 times. So if you take one of those florets, right, take this one, it looks like the whole object, right? And then if you take within that, you have another baby floret that again looks like the whole object. And then again, and again, as I was saying, you can go 7 generations like this. Okay. So this is something that is common to fractal objects. And my colleague and friend, Stephane, who's one of the co-authors, seen here with his son. Stephane, when he went to a market in Paris, um, you know, saw this broccoli romanesco. He had never seen it in his life. And he says, "This is a fractal. This is fantastic." And his postdoc advisor told him, "Yeah, but there are spirals." And so that's how Stefan started looking at those spirals in plants. And he and his advisor wrote a series of quite well-known papers. And that's how, when I went back to Paris, I'm originally from France, uh, he was the first person that I wanted to meet to talk about these things, and since then we've been friends and collaborators. So since we are at that, so this is Stefan. Um, the idea came out of a chance meeting with Nancy, who is across the river, the Connecticut River from me, at 10 miles away in Amherst. And we met at a friend's party, and she said, "Oh, what do you do?" I said, "Well, I study math and plants." I said, "Oh, this is great. I wanted to write a book. Maybe we should write a book together." And that was the beginning of our collaboration. And I said, "Okay, yeah, but we have to bring in those people with whom I'm working on this." So Stefan and then Jacques, who was at Harvard at that time. And they both helped me in creating um, a exhibit on the subject that we're talking about today, an exhibit that I put up with some colleagues at Smith College's Botanic Garden. So Jacques himself is the biologist in the group, and he now lives with his Chilean wife in Chile. And so it's a fun 3 continent collaboration. This is one of the ways we worked is, we visited Stefan in that little town where he spends his summer, and it's a gorgeous town. And this is one of the main street of the town. So that I can tell you, we were doing research in the main street of the town. And this is an artichoke, and Stefan's wife had the idea of we wanted the artichoke to turn very slowly so that we could take a film of it and then unroll it. I will show you pictures of such digitally unrolled pictures of plants. And so we didn't have the little electric gizmo that Stefan and I usually use for this purpose. So his wife said, why don't you take the Parmesan grater?" So that was the way we made that artichoke turn and took a film of it. Uh, Transition, and let's start diving into what's in the book. Um, and so all our chapters start with a little poem like this. And one thing about these poems is that you can count the number of syllabi and um, syllables. And so you have 1, 1, 2, 3 syllables, 5, 8, and 13 syllables. So it's a Fibonacci poems. So in his mirror writing, he described 4 modes of leaf arrangement. This was a first, but Leonardo crossed one out. Who can crack his code? So this is an introduction to the chapter on Da Vinci. Leonardo is Da Vinci here. Um, and so um, the fun that I had with my daughter was to decipher indeed some of Leonardo's writing, because indeed he had said he was one of the first, he was the first to say, "Oh, look, plants come in different structures, and and you better and these structures he described in just this of this little notebook, half page of this little notebook." And the rest of the page is saying, you better, artist, you better watch out. You better draw plants correctly because otherwise you'll be damned or something. Very, it was very funny to read this. But anyway, so his classification, he recognizes patterns where he doesn't talk about spirals, but the way he describes the pattern makes it clear that in the first case, which he says is the most common, is 3-2, that is like the aeonium that we just saw. And 2-2 would be when you have leaves that are come in pair in whorls of two, and then uh, the next whirl is with orthogonal to the first one, and so on and so forth. And the next one is 1-1, where you have leaves climbing up on opposite directions of one another. And then there's this mysterious fourth mode, which we really don't know what it is. Here's the drawing that he has, and here's the text that comes with it, but it's been crossed out. So my daughter and I started doing, looking at each character and thinking, oh, does it? The rest of the text was translated, so we could know what letter goes with what. And so we had to decipher letter by letter and then, do to go to Google Translate and figure out, oh, is that a word? Can that be the word? And finally, we struck on al abete, el abete, which means the fir tree. And so, sure enough, this drawing is pretty remarkably similar to the picture of a fir tree. So that was our decoding of Leonardo, and it was a lot of fun. That doesn't mean it's the truth, right? We will never know, but at least it's one possibility. And of course, we don't know what characterizes the structure of the fir, right? It's pretty erratic looking, but if you have a fir tree as a Christmas tree, you will see that actually the spirals are very clear for a young tree, and it's all in little needles all along the trunk of the tree in very neatly spaced needles, and the branches come out at some nodes of those needles and then, disrupt the pattern in some. Okay, so now jumping in time, we go in 1832. So there was one fellow in 1754 who has, some spiral words in his text, but it's not very systematic, and he does not really recognize the Fibonacci parts of it. But there's this guy Brown, together with a fellow called Schimper, who studied those spirals very, very extensively, making beautiful drawings. Brown's sister made a lot of those drawings, and she married a third fellow in their group, whose name is Agassiz, who happens to be the guy who created the Museum of Natural History at Harvard and was a total racist and a non-believer in evolution. So that's for Brown, and they saw that the angle between successive elements was related to the Fibonacci sequence itself. Not only they saw the number of spirals in those that were Fibonacci, but they saw that the angle between successive elements in those spirals was an angle of 8 over 21 in this case, which is an angle that's very close to another angle, which the Brave found a few years later. So another Fibonacci poem, Brave Soul, Bravo, Brave, the crystallographer and divergence angle genius, whose mind itself diverged and drifted far out to sea. So it's a kind of a sad story, somebody who was enormously productive and indeed impacted the field of crystallography. People still use what is called the Bravais lattices, and but plants came first for him, and in a sense helped him in the classification of the 3D light. The 3D crystals uh, came actually from the 2D crystals that plants form in his mind, that is. Unfortunately, he went at the end of his shortish life, he became very melancholic and stopped working and stopped talking, actually. So it was a little sad. So that was the angle that he found, and that angle is called the golden angle now, and it's related to the golden mean. And so he and his brother, because they wrote the paper together, thought that that was it. The golden mean, the golden angle, was the solution to everything and the reason for these structures. And it's true that if you arrange successively uh, points according to this golden angle, you will form those spirals, and they will have Fibonacci in Fibonacci numbers. But whether this is the way that plants do it is another story. And but many times you read that that's what plants do. Plants know the golden angle, and that's what they do. And our hypothesis is no, they don't need to know that. Um, so let's see. So jumping quickly, and I will come back. I will fill in. I will fill in with biology, and I will fill in with a little bit more of the math. But I wanted to give you a very broad overview of the arc of the book. And so one character that comes into play is Alan Turing, and it's a very touching picture in the middle. Um, Drawn by his mother, and that's Alan Turing when he was about 10 years old. And apparently, instead of playing field hockey with his buddies, he was more interesting in watching the daisies grow.
Uh-huh.
So that's, very touching, knowing that by the end of his life, after saving Europe of the invasion of the Germans by decoding their secret military code,
Mm.
he
Very
started
good.
looking um, at computers, and he was one of the theoretician behind the computers,
Ah,
and he was, hello? Okay. Uh, he was one of the theoreticians behind the computer, and there's something called a Turing machine, which is like a model computer before the computers ever worked. But he finally got his hands on one of the first two computers in the world, and the first thing that he programmed was trying to program plant growth, so and phyllotaxis. Um, so that's very touching. Um, So transition. So this is one of the unrolled pictures, and I hope you all recognize that this is a um, um, the word escapes me in English suddenly. Ananas is the Latin word, I believe. Um, uh, uh, Somebody help me?
Pineapple.
Pineapple. Thank you. Sorry about that. Um, yeah, so here we often use these models of flattened stems or flattened organs. And here the spirals are not spirals anymore, but they form rows, right? And then you can count them as well. And in this case, I'm counting 8 in this direction, and I believe 13 in that direction. All right. And what we see here is a kind of a change of shape of the scales of the pineapple. You see how it's like a elongated rhombus here that gets flattened and flatter and flatter and flatter. And then suddenly there are transitions, change of organs. But where we had 8 and 13 rows here, I believe we only have 5 and 3 in for the leaves of the pineapple there. Okay, so now I come to a little bit more science and try to understand what's going on and what forms these patterns. So I I leafed through my wife's master gardener manual that she used for her training, and the only things that I could see that were relevant to this talk here is a picture of the anatomy of a bud, and in particular where the apical meristem is mentioned and shown here. So it's this tiny little tip of the plant, and that's where the plant is growing. And it's a meristem. Um, when you talk about stem cells, that's what they are. They're not differentiated yet. And so the plant grows through this meristem and creates structure on its side that it leaves behind as it grows. And the structures are, scales of a pineapple, leaves, branches, etc. Right? And the other thing that is relevant to us is that I was looking for the word auxin because it's been shown only recently that auxin is one of the main player in the creation of those patterns. And I saw in that book how auxin is responsible for the curving of the plants towards the sun, right? So heliotropism. Uh, I was a little bit saddened that there was no mention of phyllotaxis. So phyllotaxis is this field that I've been talking about. So phyllo as in leaf and taxis arrangement. So, but it's a very fat book. And so I understand there's lots for people to learn in that training. So I know my wife spent many long nights studying it. So now going back in history, there was this fellow Hofmeister who was a self-trained botanist. He was destined to take over the music store from his father, but he would sneak out and do studies of plants, and he was very myopic. And so he was very good at seeing very uh, close by little elements in plants. So he was able to dissect them and study them under the microscope. So he was one of the first people to do that, and he has incredible drawings. And you could see that interestingly, the numbering of these primordia, because that's what they're called. They are organs before they become organs. They could be leaves, they could be branches again. And the way he's ordered them is through their age, right? So one would be the youngest, and between 1 and 9, there's 8, and between 1 and 14, there's 13. So, and you could see that difference going throughout those different, presumably, spirals. So, um, so here's a more contemporary picture of the same types of objects, namely meristem, again, where the plant is growing and leaving out primordia. So you see here a barely visible bump here that will become a primordium. And I'm curious if anybody has a guess of what these are meristems of.
Feel free to put it in the chat. Sunflower, artichoke,
Wow, we have a
center
winner.
center of a daisy.
So very good guesses. It is an artichoke. So it's on the right. On the right, we have an artichoke. And on the left, uh, do we have do we have a winner? On the left, it's a spruce. It's the branch of a spruce, the tip of the branch of a spruce.
No
Right?
winner for that. No.
Yeah. And pretty amazing, no? So the structures are essentially the same in the case of the inflorescence on the right. At the end, it's the end of the growth, so the meristem fills in with primordia, as we know well, right? Well, in this case, it would be the artichoke heart with all its little hair, right? Each of these primordium is going to become a hair. Um, So this picture is the one that brought me with Jacques, the biologist, because I saw that it was the cover picture of Nature or Science, I can't remember which. And I said, oh, we have to have that in our exhibit. So I contacted Jacques and he came to the exhibit and then we started writing grants together and working together. So that's how we got to work together. But this picture has been very influential to us. So let's see. Hofmeister came up with a rule by looking at those meristems, which he did over and over and over again. He discovered, and he was very enthusiastic in the text. You can read out. I mean, I don't speak German. It was written in German, but I could translate. But you see the enthusiasm. He says, I found this rule, I found this rule. And the rule is very simple, is that the new primordium forms in the least crowded space left by the others, right? So here we have the new primordium is that inkling of a bump here, and it's as far as possible from the latest one, right? It's pretty far away from 40 and pretty far away from 39, right? It's closer to some more ancient primordia. And so on the right, this is a simulation where we used just simply Hofmeister rule geometrically and dynamically to simulate the growth of a plant. And you see that the numbering is the same, the topology is the same. So it's pretty cool to see that that simple rule is enough to create patterns mathematically that espouse very closely the actual structure of the plant. Okay. So, so what about the so, Hofmeister said, I discovered this rule, but it took centuries, well, 130 years to be more exact, for people to actually discover what was behind that rule, right? And it was not until the early 2000s that people discovered that indeed oxen plays a central role in this. And so, first of all, it's transported in the sense that whenever there's a concentration of oxen, the cells are polarized and have a marker that says to oxen, go this way, go this way. That's where the crowd is. So follow the crowd. And so the oxen gets concentrated in certain spots and helps the cell growth and multiplication. And suddenly you have a primordium that is formed. But in the second step, uh, the oxen is pumped out from the primordium at its center because it's starting to create vasculature that's going to feed that particular leaf or branch or fruit. Um, so now there's less oxen around this recently formed primordium. And so the next primordium is not going to be formed anywhere close to this or to another recently formed primordia. So that's where the rule of Hofmeister is confirmed. The next primordium, here, this one, is going to occur as far away possible from the most recent ones. So that's Hofmeister rule that's been confirmed. And there were a flurry of papers from different groups who had found the same thing. So there was a very healthy and interesting competition. In 2003, suddenly three papers appeared in Proceeding of the National PNAS. So, we jumped in and started looking at these structures. And one conceptual thing that we did, because we were trying to analyze those patterns and their transitions, was to think not in terms of the spirals themselves, but what we called fronts or primordia fronts. So a front is at some point in the history of the plant, it's like the last row, the front row of the primordia, and it forms a zigzagging line. But the cute thing about this is that these zigzagging lines also account for the number of spirals. So for instance, if I count the segments, the green segments here, I have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13. So I have 13 green segments, and each of these green segments crosses one red spiral. So there are 13 red spirals, and you can count them. And likewise, there's 8 red segments, and that counts the green spirals. So these fronts are a little tool that's more localized, that's computable, that we can program, and that count at a certain time of the history of the plant the number of spirals it has, because that number of spirals can change. And we analyze the artichoke on the right here, and you see its pattern. So it's the same artichoke that we saw earlier. This artichoke, the same picture. We put little dots in the middle of each primordium, and we connected the primordia that touch one another, and we got this graph, and we counted the fronts. And we have three fronts here. The first one is 55.34. The next one is 34.21. And the last one is 21.13. So it's the all part of the Fibonacci sequence. But what we discovered is that it's a very orderly transitioning between each of these fronts. And what's involved is what we call hexagons, that is those faces of this graph that have five sides. And we see that each time we hit one of those pentagons, one of the spirals is going to die. You see, this spiral is starting here, is going to die, and whereas the other ones continue, but they are crooked. So that's when we started understanding how transitions occurred. This is a transition that goes from large Fibonacci to smaller Fibonacci. So then the next question is, how do we increase Fibonacci instead of decreasing it? And the answer is through triangles instead of pentagons. And so here's a picture, and it's a relatively rare picture that Stephane uh, you know, as we were writing the book, I said, we need a picture that shows the transitions of, Fibonacci transitions. We know they occur, but we don't have a clear picture of this full scale transition. And so one day he was at a flower shop and he saw that ornamental cabbage. And he thought, wow, if I cut the leaves of this cabbage, I bet you we can see transitions. And sure enough, we could see fronts that go from 1, 1 to 1, 2 to 2, 3 to 5, 3, and then to 5, 8. And each time we have transitions with those triangles. And so I want to mention that not everyone is Fibonacci. And so, in fact, we've done a lot of work on the non-Fibonacci examples, and we are presently and trying to understand what's the transition. So I will be talking about that, but this is an example that my students analyzed of a little strawberries that we went picking on the Smith campus. They're wild strawberries. And statistically, those wild strawberries were more like 1920, 1820, 2021, but never just sequences of Fibonacci. And so we'll talk a little bit about that. But let me give you a little bit more of a sense of why Fibonacci occurs. And we're going to concentrate on this rectangle in that picture where you see a transition occurring. So let's see. So there it is. Um, so how do these triangles occur? What does that mean? So first of all, let me explain to you. This is like, the same way that we unrolled a pineapple. This is an unrolled, abstract plant, if you want. And these circles are to denote primordia. And so we start here with a structure which has at the bottom 1, 2, 3, 4, 5 spirals going in one direction, and 1, 2, 3 going in the other direction. Okay, and and we have a front here which has 1, 2, 3, 4, 5 red segments, and that fits with the fact that it's counting the green spirals. And we have, and so why do we have, and we have 3 green segments which count the 3 red spirals. So what are those triangles and why do they occur? Well, if you look at this situation here, let's concentrate on this one, right? Uh, you see that the angle between these three disks here is too wide for the disk to sit between the two outside disks, right? Here, compared to this situation here, right? In this situation, this disk here, the inner disk, cannot go inside, right? Because the angle is too small. But as the disks become smaller and smaller, oh, that's something that I should explain why we're making those disks go smaller and smaller. I will in a minute, but let's assume that they do go smaller and smaller. The angle is too large here for this disk to be sitting on the two external disks. So it has to choose to sit on a pair of disks inside. And so the way that it's going to do that is it wants to go as far down as it can. That's the idea of being as far away from the new primordia as possible. So as far down as you can. And the way that is going to happen is if it sits on the one pair that's the most horizontal, right? So it's either the pair joined by this red segment or the pair joined by this green segment. But there are less green segments, three, than there are red segments. So the red segments are, in average, all flatter, more horizontal. So the new disks are all going to sit on these pairs joined by red segments. And so that's where we're going to get now for each red segment, we're going to get a new green segment. And so we had three green segments earlier, and now we're going to have five more. And so that's 3 plus 5 equals 8. And that's the Fibonacci rule. So this is how we go from 3, 5 to 8, 5 in this example. Okay, so I'm going a little fast here. We wrote papers about it, but this is essentially a geometric rule that implies that if you think about fronts and their geometry and their number theory, you see that one front, which is 3, 5, will beget a front 8, 5. And up it goes when the disks are getting smaller and smaller. So now I have to explain that to you, right? Why are the disks getting smaller and smaller? Well, it's really the stem that's growing, right? The stem is growing in girth. And so each primordium that stays essentially of the same size relatively to the girth is getting smaller, right? So we represented that in a cylinder, but we also have models where we represent this growth on a cone because really the stem is growing. So this is my take on it, or our take on it, and that's why, you know, why we think that Fibonacci occurs. But then the question is, what happens? Because there's a very fine equilibrium here. Do you see how, it's a close call between putting this disk here or against these two, right? It's a very close call. And so it turns out that this works if the disks are decreasing slowly, but if you start decreasing very fast, you get something different. And so you see how fast this decreases, and you have chaos, and you have triangles of both colors mixing together. And in the end, invariably, we find you have those patterns that are more, whose numbers of spirals are very close together instead of being Fibonacci apart. And so that's what we have been calling quasi-symmetric because you see those spirals, they seem to be going in about the same slope instead of the Fibonacci where the slopes are markedly different. I'm getting close to the end of my talk, and so we'll have time for questions, but I want to give you a sense of the rest of the book. Uh, no, importantly, yeah, I have to tell you why Fibonacci phylotaxis in terms of evolution. Is it light exposure? Well, some people have made models where if you change the shape and size of leaves, Fibonacci has no advantage. And what about the cacti? They have Fibonacci phylotaxis, but they have no advantages of being exposed to light. The leaves, which are really the 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 needles. So what about optimal packing of organs in a bud? Yes, that's a possible one. That's my favorite one. What about weight distribution on the stem? Yes, possibly vasculature distribution. But if you ask biologists, the kind of answer you're going to get is the following, and that I got that from an Encyclopedia of Life Science, written by an eminent botanist. So I'll let you read the occurrence of an astonishing diversity of phylotaxy pattern in a single species or even an individual, like Abies, Picea, Magnolia, points to a considerable degree of non-adaptiveness of morphological variation with respect to phylotactic patterns. So it looks like, plants will do what they do, and one single plant might adopt Fibonacci and another one might not. The same tree that Stephane looked at in his village that I showed you earlier, same pine tree, one year had all Fibonacci, 99% Fibonacci. The second year, 30% were non-Fibonacci, 30% were quasi-symmetric. He asked the farmer, what's going on? The farmer said, oh, they cut the tree, the branches uh, along the road. And so the tree was stressed and it changed the parameters in its growth. It started growing faster, maybe. And then suddenly it was off in a quasi-symmetric range. So in short, why evolutionarily do we have Fibonacci? No one knows. There's no real consensus on this. And so, which makes it really interesting. My take on it is that, it's a relatively simple, robust system and which is adaptable to many different situations. And maybe that's all we need. And but the patterns can be very different. Certainly, Fibonacci is not the unique answer, but it's a logical answer if you're growing with a relatively slow growth. Um, that's a beautiful example of non-Fibonacci, even though it looks pretty regular, but it's not Fibonacci at all. So the rest of the book, um, and I will, give you a few minutes for questions. We did look at fractals, and we had a discussion, Stefan and I, what constitute fractals. Mathematicians are nitpickers, and we have very precise definitions that are not very relevant to nature. Um, leaf Kirigami, a student of Stefan, actually wrote a PhD thesis about how uh, the way that leaves forms are created is often when you have two leaves in a bud, and you can think of that as crumpling some tissue and cutting in half. And that's exactly what kirigami is. Cell division is a beautiful thing here where Jacques created a model of cell division according in the same way that bubbles are divided and came to three or five possible fixed patterns, and this is one of them seen on the tip, a meristem of a fern. And then at the very last minute, our editor said, "Oh, you have to talk about animals." And we said, "Well, there's no phytotaxis in animals," but we did our best to assuage them, and we found interesting things. You know, the eye of a fly, for instance, is created in a way with fronts of cells, but not with the same topology of a cylindrical topology of a flower. And then finally, we have fun. There's some activities at the end of each chapter. You can have hands-on activities, and the final chapter is full of recipes with plants that exhibit Fibonacci, starting with a pina colada. So I think that's it. Any questions?
There were not questions. There's a lot of comments. Beautiful. I bought your book. Thank you so much for joining us. I am fascinated by Fibonacci sequences in nature. I hope you will describe any advantages these structures confer on plants or other. And that is it.
Okay, cool.
My favorite part of the book is Alan Turing's work. And I'm waiting to see if any other questions come in. On Oh, and Yogesh, are you here?
Yes, I'm here, John.
There's a comment on on the survey form. It wants a
Yeah.
number instead of a name.
Well, it came from, um,
And Chris, the last comment is also bought your book, gorgeous and fascinating. Thank
Yeah,
you.
now I'm looking at the
Yeah,
comments. Yeah.
yeah. So super nice talk. I'll see if Yogesh can figure out the form. And
yeah, it
then,
The
came from
form which
Jennifer.
yeah, I don't know. But we'll be back in 2 weeks talking about irrigation.
very good. Thank you, Yogesh, for the invitation.
Professor
We
Gold, I thank you so much. Thank you so much. I
we
really appreciate it.
Yeah.
we were wondering earlier how you came across my name or anything.
Well, the book, for
Okay.
starters,
Yep.
yeah, I've had the book for a few years now. Very much
Okay.
enjoyed, and it has all my favorite names: Mandelbrot, Turing.
Uh-huh.
Uh, so yeah, So I was looking forward to this, and yeah, thank you so much.
Cool.
Joan, can
Excellent.
I ask one quick question to
Yes.
clarify? So, Dr. Golay, you mentioned a few things that were posited about and advantages.
Yeah.
Nothing is agreed upon in plants or pine cones or anything. I mean, you mentioned a few
Yeah,
possible.
no, it's the amazing thing that yeah, you ask. I've asked quite a few. I'm not a botanist myself. I'm not a biologist, but I've asked quite a few, and they essentially all had their, you know, we don't have a clear answer, and but
Okay.
each had a little preference. Some had a preference for, oh, if you can pack as many um, as many organs as possible in the bud, that's an advantage, right? Because you don't have to use as much energy to protect it, right?
Hmm.
Because they're closely packed. Um, but even that was, and it is true that if you use Fibonacci phytotaxis, it is optimally packing
Hmm.
things, but it's not the only optimal packing. There's many other pairs of numbers which will give you an optimal packing in the sense. And so, and then you have plants that
going to be that way as we head deep into the afternoon. And
uh, there are plants that have Fibonacci on some side of the tree and then not Fibonacci on the other side. And so then suddenly, the evolutionary advantages are not so clear anymore. But maybe so Stefan has this hypothesis that it's actually, we are in a range of regime which is right between Fibonacci and quasi-symmetric, and being able to switch between one and the other might be an advantage, right? And so, but it's fascinating how little is agreed upon at this point.
Okay, I guess that's it. Thank you so much. Stay safe with that storm coming.
Thank you.
Yeah. Thank you, everyone. See you again in 2 weeks. And if you can click on that link, you can put your number and then an email. Okay. Anything else, Yogesh?
Thanks, Professor Go. Really appreciate it.
Okay. All right. Bye,
Thank you.
everyone.